Thanks for previous two talks sharing, and I have learned a lot. I'm trying to give more new ideas in this afternoon for drug discovery. I'm Zuolong from Henan University, and my supervisor is Chen Xinbo. And today's topic is to predict drug-targeted binding affinity by using deep learning methods. Uh, as we all know, drug development is very time-consuming, and all of the computer-aided methods or AI-assisted methods are focused on the preclinical research. Among them, drug discovery and optimization is the most challenging part. So the drug discovery is the topic of this work. We want to predict the drug target binding affinity. There are three approaches can tackle this problem. Molecular docking is familiar to everyone. It simulates the docking process in physical level. Machine learning and deep learning based methods are all relying on the affinity data size, and then they can to predict affinity quickly. There are three challenges, but after today, I see that the fourth challenge has been solved. So only three challenges. The third challenge is the number of the molecules and proteins in the affinity data size is extremely small. So if the model only trained on the affinity data size, they cannot get more powerful representation of molecules or proteins. So we want to use the pre-trained model that is trained on the large unlabeled data size. And in this way, we can get more powerful representation of molecules or proteins. The second question is that existing methods often ignore the significance of subgroups of proteins or Pharmacophore. So we want to incorporate the substructure of molecules information in the model. So in this way, we can to improve the performance of the model. The third question is that state methods fail to capture the interaction level structure information. So we want to introduce a novel feature fusion methods that can make the model to learn the interaction level information in the training stage. This is the overall architecture of the model. For molecules, we first get the pharmacophore graph, and then use graph neural network to learn its pharmacophore level information. And after that, we use Radkit to get the atom bound graph, and use pre-trained model named Jim to get node feature of each atom. And we use some chemical information to initialize the edge feature in this graph. Then we use graph neural network to learn the atomic level information of molecules. For proteins, we first use pre-trained model named ISMB to predict the residue content map of protein and residue animal acids as nodes, and predict contact probability and edge weight. After that, we use graph neural network to learn the graph level information of protein. And in the sequencing level, we use the pre-trained model to get the node feature of each animal acid, and then we can get the evolutionary information of the sequence. So we use some FCN network to get the sequence level information of protein. In the feature fusion block, we can get the drug target pair representation. In the final stage, our model can predict the interaction probability or binding affinity. We use the two pre-trained model. For proteins, we use the GEM. It is developed by the pedal helix, and it uses 3D structure of the compound, includes the atom bond graph and the bond angle graph. From protein, we use the ES1B. It is developed by the Meta. Firstly, we use it to predict the residue content map of protein, and secondly, we use it to get the node feature of each animal acid. Pharmacophore is one of the contributions of our work. Take this compound as an example. Firstly, we decompose the 2D structure, and then we can divide the structure into several substructures, and we will visualize every substructure to a virtual node in the pharmacophore graph, and we will virtualize an edge between each substructure. After get the pharmacophore graph, we use graph neural network to learn the pharmacophore level information. And this is the feature fusion block we proposed in our work. And by using this block, we can we can make the model to learn interaction level information in the bending precise between molecules and the proteins. In the experiments, we use 4 data size, K-bar Davis human elegans. As you can see, the number of the molecules and proteins in this data sets are relatively small. So we want to use the pre-trained model to make the model can get more powerful representation of molecules or proteins. And the ratio between positive sample and negative sample is close to equal. So we want to test the robustness of each model in different ratio scenario. The baseline method includes machine learning baseline methods and deep learning baseline methods. We test our model on 3 tasks, including regression tests, classification, and clustering tests. In the regression test, our model achieved the best performance on both data size and on every evaluation metrics. And we also do the average study to see the contribution of each 3 core structure in our model. And we can see by integrating all of the 3 core structure at the same time can maximize the performance of our model. We also test the performance on the classification task. We also better than other baseline methods, but if the size of data size is larger, the difference between our model and other baseline methods will be further amplified, or the advantage of our model will be more obvious. We also test the model on 3 code scenarios. Take a code protein as an example. The proteins in the test data set are not present in the training data set. So the code pairs scenario is the most challenging part. From the bar graph, we can see the heterogeneity achieved the best performance compared to other baseline methods. We also clustering all of the drug target pair into the in-binding space. Includes the positive sample and negative sample. As you can see, the results given by heterogeneity, the results is more compact. There's a clear line between positive sample and negative sample. In this table, we can see the heterogeneity can get better results on the clustering in matrix on 2 data size. So the 2 experiments shows that our model can be better than other baseline methods on regression tests, classification tests, and other code scenario, because our model in the in-binding space can distinguish the positive sample and negative sample more clearly. So it can be explain why our model is better than other methods. In the case study, we first pre-train a model on the K-bar data size, and after that we use the trained model to do the drug screen. We have the component library. It contains about 300 molecules, and the target protein is the M-Pro. And we use the trained model to try to find some potent molecules from the component library. In the library, it contains about 10 known inhibitors that can to inhibit the activity of target M-Pro target. From the results, we can see the results given by heterogeneity. There are 6 known inhibitors are in the top 10 results. So our model is methods in the case study. We further clustering all of the active molecules in the by using their fingerprints in this figure. And you can see all of the active molecules have the similarity. It's small, and they are far apart from each other. And you can see the red figure. In the top 10 results given by heterogeneity, there are only 2 groups of the molecules have a similarity larger than 0.5, but other agent molecules have a similarity less than 0.5 or 0.1. We also visualized the core structure our model focus on, and you can see the core model our model focus on is for overlap or partially overlap with the known active sites. So you can use this heat map to further optimize the structure of the molecule. So we want to use the transfer learning and data augmentation and other tech to enhance the model performance. And more importantly, we want to integrate heterogeneity with other traditional methods to advance drug development. So we want to discuss 3 questions. Is how do we develop the prototype based on the research, and how do we turn our research to a practical platform? So in the second year of my master degree, and I have developed a platform that is called Smart Drug Finder. It is a cloud-based smart drug screening platform. And in this year, I have collaborated with some students from School of Pharmacology and the School of Business to hand find a feasible business plan for this platform. This is the core process of our platform. First, we will collect some open source small molecule database, and then we will do some pre-precise to this data size. After that, we will use artificial intelligence to do virtual screen. And in the third part, we will have solubility-based screen stage. And in the final stage, we will do molecular docking and web test to finally find some potent active hits. This is the technical detail of our platform. We developed and deployed our platform on the Huawei cloud, and all of the data in the platform are stored in MySQL and Redis. And after user logging in the system, you can use some service like drug discovery, drug repurposing, affinity prediction, and other service. This is the development vision of our platform. In the technical base, there are some hardware, GPU, CPU, cloud resources, and deep learning-based methods. In the system platform, it is our smart drug finder. This platform includes some models like laboratory processing, virtual screen, multitask scheduling, and the docking and kitchen mechanism can make the platform running more smoothly. In the service, we have three services: drug discovery, drug repurposing, customize the service. It means that we can to develop more powerful models based on the needs of the users. And in the pipeline project, we can do our in-house pipeline, or we can to collaborate with other big pharma or CRO company to do research. And in the running of the platform, we can to accumulate our in-house data. So there's a good feedback in our platform. This is some screenshots of our platform. This platform is Chinese language, but we will upgrade in English language. But now tasks cannot be executed now due to high cloud GPU costs because I'm a student. I cannot afford the GPU fees. I'm very thankful my supervisor, Chen Xinbo, and I come from Hainan University, and I thank all of my colleagues in my lab. I'm seeking a 2025 for PhD position in for medicine, so I'm very happy to talk with you guys. And so that's all. Thank you. Thank you.
Let's thank our presenter. We have a question here.
I'm curious to learn more about the aspect that you mentioned as a challenge, insufficient data. So I was wondering, like, what is the scale of data you are referring to at this point? And also, hypothetically or in theory, what would be the scale of data you would consider to be sufficient? So is this a general?
You mean sufficient data?
Yeah, so you mentioned that current data is insufficient.
Oh.
So I was wondering, like, what is the scale of the data you have that you are considering insufficient? And what could make you happy? So if you
I'm not sure I understand your question, but insufficient data because drug target binding affinity data is limited in the paper or in the open source platform. In most scenario, we use several data sets, and it only focus on some target proteins. We can have a data set and have several various proteins and their interaction with several unique molecules. If we have this data set, it's very good. There's a Kaggle competition in this platform, and they want to tackle this problem because they publish a data set is very large, but the target protein is limited. It's about 3 target, but they have molecules bands to them about 3 million. So it is useful, but it's like use some best methods. Thank you for the question.
Cool. Do we have any other questions for the presenter? I think when it comes to how much data is enough, particularly, I think it becomes a philosophical question given the size of the chemical space that is druggable. So it's estimated to 10 to the 60 molecules. So it's never enough. It's all a drop of water in the ocean. But I think something to keep in mind is how you select molecules that will most benefit the models. Have you thought about any clever active learning selection methods that could then, in a loop, generate more data and then improve the models over time?
Yes. After learning, I have an idea. Is that we can to use the deep learning methods to predict unknown molecules and the target, and after that we can to minimize the scope of the library, and after that we can use docking, because docking is more accurate than deep learning based methods. And by using docking, we can to get more accurate affinity. After that, we can use this data to retrain our model, and then there's a good loop in there.
Cool. Thank you.
Thank you.
All right.